Hi, my name is Shah Faisal. I'm a biotechnology graduate from Bacha Khan University, Chasada, Pakistan. With a passion for scientific innovation, I've published 35 research papers in peer-reviewed journals, collectively holding an impact factor of 183. My work has been cited over 2,160 times, with an H index of 23 and an I-10 index of 27, reflecting the depth and reach of my contributions to the scientific community. Currently, I'm a researcher at the Guangzhou Institute of Biomedicine, and in this video, I'm going to teach you how to design a complete biomedical research project, from scientific discovery to product commercialization, publication, and even patenting. Let's dive into how I approached one of the most complex challenges in liver disease, NAFLD, or non-alcoholic fatty liver disease. I began with transcriptomic analysis. Using RNA-seq data from liver biopsies, both NAFLD patients and healthy controls, I performed differential gene expression analysis using DSEQ2. This helped me identify DEGs, or differentially expressed genes, that were dysregulated in NAFLD. I filtered the genes by adjusted p-value and log2 fold change, focusing on the most statistically significant ones. Then I used functional enrichment tools like KEGG, DAVID, and Enricher to uncover which pathways these genes were influencing. The most affected processes included lipid metabolism, inflammatory signaling, oxidative stress, and fibrosis, core hallmarks of NAFLD. Out of hundreds of genes, one caught my attention. Let's call it Gene X. It was upregulated, pathway-enriched, and poorly studied in liver disease, making it a promising novel candidate. To validate Gene X, I moved to the laboratory phase. I extracted RNA from NAFLD mouse liver tissues and performed qPCR to confirm its expression. Then I used Western blotting to validate protein-level changes. Expression was elevated, just like the transcriptomics had predicted. To visualize localization, I conducted immunohistochemistry, IHC, on liver sections. Strong staining appeared in hepatocytes and fibrotic regions. I followed with immunofluorescence, where I observed co-localization with immune and fibrotic markers like F4/80 and ASMA. That confirmed Gene X as a driver in disease progression. Next came the therapeutic phase. Using AlphaFold and Swiss model, I predicted the three-dimensional structure of Gene X's protein. Then I analyzed potential binding pockets using Cas-TP. I conducted virtual screening and molecular docking with AutoDoc-Vena to find small molecules that could bind and regulate this protein, either inhibiting or enhancing its function. Top candidates were subjected to molecular dynamics simulations using GROMACS. The most stable and high-affinity compound, Compound A, was chosen for further testing. ADMET predictions showed it was likely to be safe and orally bioavailable. We synthesized Compound A in the lab and tested it in vitro on HEPG2 liver cells. The results were promising reduced inflammation, downregulated Gene X, and suppression of downstream cytokines like IL-6 and TNF-alpha. Next, we moved into in vivo validation using a mouse model of NAFLD. After treatment, liver histology showed less fat accumulation, reduced fibrosis, and decreased inflammatory markers. From here, the project shifted toward commercial and translational goals. We completed preclinical toxicology, filed for a composition of matter patent, and prepared an investigational new drug IND application. We partnered with a biotech accelerator to begin clinical trials. Phase 1 confirmed safety and optimal dosage. Phase 2 tested efficacy in NAFLD patients. Based on strong results, we plan to scale to Phase 3 and eventually seek regulatory approval. In parallel, we prepared and submitted multiple manuscripts to peer-reviewed journals and filed intellectual property for both the compound and its target pathway. This entire process, from omics to drug design from bench to bedside, is a model for how to structure biomedical research for real-world impact. It's not just about publishing papers. It's about creating cures, filing patents, and translating science into market-ready solutions.